Hello, how are you guys doing? Um, Welcome to the microbiology lecture course. I hope you have fun with me here. I always have fun when I'm teaching this course because I think everything we talk here is so interesting and so enriching, and I think you're gonna see that. So we're gonna start every video that I record to you. I'm always going to show you the first thing, the learning objectives for that chapter. So if you want to know exactly what you need to know to do well in the exam when it comes to each chapter, you need to look at that Word document that I posted in iCollege in the Start Here folder that is called Learning Objectives. Okay, so these are the learning objectives for this chapter. We're going to learn the binomial system of nomenclature, the difference between the microbes and what microbes are, and the importance of Louis Pasteur's experiments and his work in microbiology. You're going to see a lot more than this in the chapter. You don't need to know more than this, okay? Cool. So let's start about talking what are microbes. So anything that is not visible with the unaided eye, that you need a microscope to see, it's called microbe. So what's included in this big category? Bacteria, fungi, protozoa, algae, and viruses. Okay? And you're gonna see when we talk here about diseases and about the organisms, you're gonna see helminths. And then you're gonna say, but helminths are worms. And they are included here because their eggs, and most of the time their larvae, are microscopic. That's why we also study worms in microbiology, other than these groups. Where's my pen? Other than these groups that I mentioned here. Okay, cool. So let's think about this. I want to change where I put this because I want it to be able to see everything. Okay, so some microbes are pathogenic, right? So we know this. This is, where's my pen here? I'm learning this new pad thing, so please be patient. Some of them are pathogenic. They can decompose organic waste. When we talk about microbes metabolism, we're going to talk about this decomposition. They generate oxygen by photosynthesis, some of them. They can ferment. They can produce chemicals like ethanol, acetone, vitamins, okay? And they can be used in the industry for reasons, for this type of purpose. Remember that also microbes are involved in food spoilage, and you can use them to prevent disease. And if you don't really understand how this goes, we're going to discuss a lot about this in this course, okay? How they prevent disease and how transmission of the disease happens to prevent epidemics and pandemics like the one that is happening now, right? That is preventable. So let's start by talking about how we define, how we name these microbes. So we're talking now about scientific nomenclature, okay? So this was established in 1739 by a scientist called Carolus Linnaeus, and he defined this binomial nomenclature for microbes as it's like if they have a first name and a last name. So the first name is always the genus, and the second name is always what we call the specific epithet that then gives the name, the binomial name for the species. You're like, what are you talking about? So let's define this. So these names are usually originated from the Latin, and they're always italicized or underlined. For example, if I'm writing here, so I'm gonna write, let's think about the name of a microbe, amoeba produce. And I'm handwriting, so how can I handwrite and italicize? Not possible, right? So what do? I underline it. What if I'm typing that name? Then I can underline or italicize. It's more usual to see it italicized, okay? So let's give examples here. So look at this one. Escherichia coli, which we usually call E. coli. So would it be correct to abbreviate the genus and say E. coli and either underline or italicize? Yes. But you see how the name here is written in italic? Done. That's what you have to do. Genus, capital letter. Oops. Specific epithet, lowercase letter. Is this important when you're talking about this nomenclature? Totally. You need to remember this, okay? And remember that the names sometimes come from something that you can relate and help to define or identify the bacteria. For example, E. coli. The name of the scientist that discovered E. coli was Theodor Escherich. So Escherichia comes from his last name, right? Coli because they come from the colon, from the intestine, okay? Example, Staphylococcus aureus is another bacterium, right? Another bacterial species. So if you think about that, think that you have staphylo is the Latin word for spherical cells that are grouped together as little, how do you call it? like a grape cluster, okay? And aureus is because they are gold color, okay? So that is the Latin word for yellowish or yellow, strong yellow color. And so on. So that's the nomenclature you need to know. So now let's talk a little bit about these microbes. So I told you that we have bacteria, fungi, protozoa, some algae, some algae are microscopic, some are not, viruses, and multicellular animal parasites like the helminths. So here we include mostly, helminths. I am still learning this, so it's not gonna look pretty all the time. One group that I did not mention earlier is the archaea. Archaea are similar to bacteria, but they're not bacteria. Okay, so let's see how we can basically give the main features of each one of these groups. So these are some pictures of these organisms, or some of them, that we are going to talk about now. So let's start with bacteria. So bacteria and archaea are the groups that are prokaryotes, and I'm going to show you how that classification goes in a minute, okay? So they are prokaryotes. That means they do not have a nucleus. So that means no nucleus and no organelles. Okay, so we're going to talk more about this as we go. They are all single celled. So if you see bacteria grouping together, is many bacteria, many individual organisms grouping together, which makes a cluster, and they can make a colony, but each single cell is one organism. They have cell wall. Cell walls are protection for organisms that don't have skin like we do. So when we talk about cell wall, we say, what is that cell wall made of? And that's an important feature to define different groups of organisms, right? So when we talk about bacteria, we know their cell wall is made of peptidoglycan. They divide by binary, and we are going to talk about peptidoglycan, which is a type of sugar, in the next chapters. They divide by binary fission, which is very similar to mitosis, except that here we don't have a nucleus, so you call it binary fission. Okay, so one mother cell makes two daughter cells that are exactly the same size. Okay, and they can derive nutrition from organic or inorganic matter, or they can do photosynthesis. Okay. And this is the picture of a bacteria or a group of bacteria, right? This is probably, you see how this is one cell, and this one cell is called Diplococcus. Okay, there are two diplobacillus. So there are two bacillus. You know this rod-shaped cell here connected to this other rod-shaped cell together. Okay. Detail. Always look at the scale before I start talking about archaea, because the scale tells you how big these organisms are. So these, anything that is the size of this line in this picture is 3 micrometers. Think about how small they are. So they're actually smaller than 3 micrometers. See, that's the size of a bacterium. What is a micrometer? Is 10, let's say 1/90 smaller than a meter. So it's very small, okay? Okay, so let's talk about archaea now. So archaea are also prokaryotes, and they don't have peptidoglycan cell walls. They have another substance instead of peptidoglycan on their cell wall that is called, because another name for peptidoglycan is murine. So archaea have cell walls made of pseudomurine. Okay. They usually live in very extreme environments, so they're not usual pathogens of the human body. You find them in regions where you are rich in methane, because they're the ones that produce the methane. So they can produce this gas, methane. I hope you understood that this is a TH. They can be extreme halophiles, meaning what? They would love and stand high concentrations of salt. And they can be extreme thermophiles. They can withstand very high temperatures. Okay? So these are characteristics of archaea. And the reason why they are different and separated from bacteria as a different group is that one of the reasons is that they have a different cell wall. Okay, and also sequence of messenger RNA is useful in classifying these organisms in different groups. I'm going to talk about that later. Okay, so we're done with just general features of prokaryotes. So now let's talk about eukaryotes. Eukaryotes, so what do we have here? Eukaryotes do have nuclei, right? And do have organelles. So organelles are any structure that is wrapped in a membrane that looks like the plasma membrane. So that is made of phospholipids, okay? So it's made of a bilayer of phospholipid, just like the plasma membrane. Cell wall for fungi is made of a protein called chitin, not of sugar, okay? Chitin, this is the same protein that you find in exoskeleton of insects. That same protein. You know when that insect sheds that crust or that exoskeleton that you can call a shell or a crust, that is made of chitin, same protein. So fungi have their protection as this chitin cell wall because they also don't have any skin, right? And they can absorb, so this is how mostly the fungi live. They can degrade and absorb organic matter, and that decomposing function is essential for the environment. Because think about, if anything that is dumped in the environment doesn't get decomposed, we would be in trouble, right? Because you wouldn't be able to recycle these molecules. So we can recycle them mostly thanks to fungi that can decompose them. A lot of bacteria can also do the same thing. Yeasts are the fungi that are unicellular, and molds and mushrooms are the fungi that are multicellular. Okay? So molds are usually masses of mycelia, or this little hyphae that we're gonna talk about, and we will really detail the structure of fungi later on. But this is how fungi look like. Okay, so you have, this is the spore, and these are the hyphae. Okay, protozoa, also eukaryotes, always unicellular. Is there anywhere here that it says unicellular? Free living? No, it doesn't say, so let's write it down. Unicellular. Okay, so don't forget that is an important feature of protozoa. So they also have nuclei and organelles because they're eukaryotes. They can absorb and ingest chemicals as, you know, their nutrients. They are usually motile. When we talk about fungi, I forgot to mention they're not motile. They're usually like a little plant that has a little root, and they just stay there, right? If it's a mold. But even the levedura is in my language. Even the yeast, they are not motile. But when you think about protozoa, they can move through pseudopods, cilia, or flagella. Okay? They can be free living or they can cause disease, depending on how much or how independent they are. The more dependent, the more pathogenic, because then depend on the host to survive. They're not doing that to be mean to you. They're doing that because they need you as a host to survive. It's our function just to get rid of them, right? And they, that's a food particle, and that's probably an amoeba emitting pseudopods to be able to ingest, let's call it ingest, or phagocytize that food particle. Okay? Trying to look at my timer here. Okay, guys, so algae, we're not going to talk much about algae. They're also eukaryotes, cellulose cell walls, found in freshwater, saltwater, and soil. They do photosynthesis and produce oxygen and carbohydrates, okay, because they do photosynthesis, okay? This is how algae look like. So, in this green, if you're curious about what is this green color here, this green color is the chlorophyll that makes them green, because they use the chlorophyll to undergo photosynthesis. What about viruses? Viruses are acellular. They have DNA or RNA in their core, and they have a protein coat, like a capsule around them, okay? They can also have a lipid envelope that comes from the host, and we're going to tell you how they steal that from the host when we talk about viruses. So they can have a lipid envelope, and they only replicate inside living cells, okay? They cannot replicate out of cells. And this is how viruses look like on top. This is an HIV virus on top of CD4 positive T cells. The multicellular parasites are also eukaryotes, and they're the parasitic flatworms and roundworms. So these are all called helminths, okay? Some of their stage lives are microscopic, that's why we study them here. Okay, last thing I want to show you is, and then I'm gonna stop here and record a new video to talk about Pasteur. This is the 3-domain system, okay? So we have 3 groups that are here. And so these groups are called domains, okay? So 3 domains, which are bacteria, archaea, and eukarya, okay? So what do we need to know? And I just want to make sure you read here. So let me write again. So what do we need to know about these three groups? Bacteria. There are different species of bacteria that are all phyla from this domain called bacteria. Archaea, different cell wall. The RNA, so remember to go back and look at what RNA, ribosomal, there is a little R here, what ribosomal RNA means. That's homework, okay? Go look into that. So the ribosomal RNA sequences are similar among organisms inside the same domain and different if you compare different domains. So the rRNA sequence in bacteria are different than the rRNA sequences in archaea. That's why these domains are separate. And that's why you have a whole domain just for the archaea, while the eukarya, which are fungi, all the protozoa, all the helminths, they're all divided here, plants. So this is the tip here, like how can you classify all this together? So I'm going to give you a tip. Let me get a different color here. I'm going to highlight the phyla here. So one is animal, the other is plants, and the other is fungi. And you're like, okay, what about the other guys? All these other guys that are not highlighted, they belong to a separate phylum that is called, let me get another color to tell you here what all these that are not highlighted are called. They are called, it's the phylum Protista. Got it? So anything that is eukarya, not a fungus, not plant, and not animals are protista. What is included in this group? All the helminths are in this group, slime molds, some algae. Okay? Good? Actually, all the algae, because the algae are not in any of the other groups. Cool? Okay. So I'll see you in the next video where we're going to continue chapter 1 talking about Pasteur and the first discoveries of microbiology. Bye bye.